Yes, I mean, I still got there now.
Then let's continue with EEG. And the big topic for the next maybe 3 or 4 hours are event-related potentials, which is a basic set of phenomena that one can reliably record with EEG. And if you remember what we did in the fMRI course, we actually did not really talk about a set of reliable results that one can record with fMRI, although there's definitely a set that one could look at. So for example, hemodynamic responses in visual cortex to, let's say, checkerboard stimuli is something that you always get in fMRI. So in principle, there could be something like event-related hemodynamic responses, and then following the outline that we go over here also for fMRI. Some of it's not established, which is actually a little bit weird. It should become established. That would be nicer, because you basically the same phenomena that we are discussing now for EEG, where you give a stimulus to the participant and then record a brain response and then average over many representations of the stimulus. You can do exactly the same thing with fMRI. Good, but in EEG, these things exist and have been described, and it's important to actually know them and get to know the language, because EEG research does many advanced things which usually build on the basic event-related potentials that essentially, I think EEG researchers would say that everyone can record and that have been described a lot. So it's some terminology to learn and associate something with. And it's also quite interesting, like everything. Good. So before we get started with the actual event-related potentials, I want to briefly go over again what we discussed in terms of the neural origins of the EEG, because this is really also what is the underlying theory of where the ERPs come from. So remember that we talked about cortex, and that we said the cortex has different layers, and then there are cells in these layers. And one important cell type are the pyramidal cell neurons. And then we discussed this thing that if a synapse becomes active at these dendrites, then positive charge moves in, and the external milieu will get a little bit negative. And this will essentially lead to a negativity in this at this point in space, and which has to be basically equaled out somehow. And then the idea is that this can happen by internal currents inside these cells, which then relate to the fact that you get a surplus of positivity at distal points. The whole thing then leading to what is shown here on a small thing on the right, leading to a patch of cortex surface where you have some, for example, negativity on the upper part or the uppermost superficial layer of the cortex, and a positivity on the lower part of the cortex, or going to the white matter. So you should think that here, where I now, you can't see this. That here, this is moving away from the brain. This is where white matter is. And so this is the folded cortex. Here you have a sulcus. Then you have a patch of cortex like that because there are these pyramidal cell neurons which are all aligned in the same way, and the idea is then, of course, that they all get simultaneous input from outside of the brain, specifically if we are in primary sensory cortices, so they are all driven simultaneously by by afferent axons from outside the brain, for example, from the retina, which in turn then is conceptually modeled as a dipole, so something that has a positive and a negative pole located at a specific point in the brain, which then extends its electrical field and the currents that go along with throughout the skull, so that when we have an electrode somewhere here, it will become negative with respect to an electrode that we have here. At the time, of course, this whole thing is dynamic, as we will see, at the time that this dipolar activity exists. And when this ceases to exist because the stimulus is long gone, then of course this goes away. So that's kind of the simple neural origins model. I'm not saying that's the greatest model ever, but it's what people have in mind when they think about how event-related potentials are generated. And of course, there are many unknowns and unknowns in this whole thing, and it's actually a whole research field to understand the neural underpinnings of EEG better. So I think this kind of model here is intuitively kind of reasonable, but then you can of course ask the question, how many cells do you need to be active? How much cortex does this correspond to? And then the answers are usually either textbook style, they are like, you need a patch of like 2 by 2 centimeters, something like that. But where do these numbers come from? Well, they come then usually from simulation papers, and there are not that many simulation papers. So if you're really interested in the neural underpinnings of EEG, it's an interesting topic. Might actually want to do a master thesis on this. I would be happy to supervise that. It's interesting. And we obviously do some modeling of EEG responses in our lab. But the unfortunate, so that's the positive view. The negative view is we don't really know where these event-related potentials on the scalp come from. Same for fMRI. This is why it's interesting. This is why I always say there's no real answer. There's no gold standard, because whatever you record, you don't really know what's coming from. And then what the real challenge and interesting thing is to build models that try to explain where these signals come from. And then have a theory about what these signals mean, because you build the theory yourself. Good, so that's the simple model. Not the greatest ever, but a model. And useful. And then we also discussed already how the standard EEG experiments work. So you have the electrode cap on the participant, and the participant is exposed to some stimulation, for example, visual stimulation using a monitor. And then you look at or you record continuously the activity of a single electrode over time while stimuli are given, in this case these X's and the zeros, and these happen again over time, and we will look more closely at how you set up this and the whole setup, and we will look closer at in later stages. And then the idea is of event-related potentials, that you know the time when a stimulus was given, and then in the post-processing of the data, you can cut out this respective time points, which then leaves you for a specific electrode following a specific event code, leaves you with these traces, and then you can average them together. For example, as shown here from the log book, the average of 80 of the X's and 20 of the O's, and then you get something that you would call an event-related potential, which has some peaks and valleys, and these peaks and valleys then, as we also already discussed, get some labels: P1 for positive peak, the first one, N1 for first negative peak, and so on. Yeah, so this is kind of where, without going into the details of the data processing, which we will do later, but where essentially the event-related potentials come from. Yeah, good. And now the idea is to go over the zoology of event-related potentials. And a first big group of event-related potentials is related to primary sensory events. And the typical ones that you see around are visually evoked potentials, somatosensory evoked potentials, and auditory evoked potentials. All of them because you can precisely and easily create stimuli in these domains using, of course, for example, a computer monitor or headphones or electrical stimulation or mechanical stimulation of the skin. These are sensory event-related potentials that you find a lot. You can, of course, also, or people try at least, to, and I think have successfully tried to record potentials in response to, for example, gustation or odor. I think in Potsdam there's actually a group that does that. I don't remember. What's her name? There's a female PI, or a woman PI, I should say. Woman PI, and I don't remember her name, but she's at the University of Potsdam and does odor EEG, I think. One of your colleagues, Sarah Kolb, she did a master thesis with her. So one can do more than these visually, somatosensory, and auditory, but visually, somatosensory, and auditory are simplest. And I'm mainly using these days visually evoked potentials because they are the easiest of the easiest. There are not only sensory event-related potentials, but also what I call here cognitive event-related potentials, which are, of course, for cognitive neuroscientists, maybe of even more interest than the sensory event-related potentials. Namely, well, they are more than these four, but these are ones that we discuss. Maybe if I manage, I put in one more. Which are mismatch negativities, P300, language event-related potentials, and error-related negativities, all of which will become clear when we discuss them, are somehow related to, let's say, higher level surprises or changes in something. You will see that all of the event-related potentials that you see on this slide, they relate to that something in the outside world changes. For example, a visual stimulus comes up, a somatosensory stimulus comes up, auditory stimulus comes up, and here it's something that changes in the environment with respect to what has happened previously. So my general model of what happens or of neural activity in general is that it's the neural activity measures changes, and which makes some sense in terms of metabolic costs. So if nothing changes, why should you waste metabolic costs? But if something changes, then you might want to signal that. I will come back to that. The final thing that I'm actually not that familiar with, but should get more into now that we're doing actually the decision from experience EEG, are movement-related potentials. These are potentials that you see in the build-up to movement, and we will also discuss that. So that's the entire scope of things that we're going to discuss, and this part of the course is fairly straightforward. We go over them one by one in kind of a structured fashion. So it's relatively straightforward for me at least, to present it. I don't know whether it's easy to follow, but it's also definitely relatively straightforward to memorize because it's always the same kind of structure. And as long as whenever there's structure, things get simpler. So we're starting with visually evoked potentials. One standard paradigm to record visually evoked potentials is the response to what is called a checkerboard. Like many things in cognitive neuroscience, the terms, or maybe even beyond cognitive neuroscience, the terms don't really make sense. Like we discussed last time that the term local field potential doesn't make sense because these are just potentials. The term checkerboard, these are chess boards. I don't know why they are called checkerboards, but these are chess boards. So these are tiles of black and white tiles. So make it a little bit bigger. So here you see first one checkerboard, and then you see a reversal, which you can only tell by the this middle line here, where actually everything that was black turns white, and everything that was white turns black. And these are presented to a participant at time 0. It's just a checkerboard, and then it flips once after 500 milliseconds. So you see it, so it's not really movement or something, you just see it coming up, and then it changes after a little bit of time. And what one then observes is here shown for electrodes on the contralateral side of the cortex to the presentation of the checkerboard. One observes a visually evoked potential to a checkerboard, and this has kind of a very reliable and reproducible and reproducible shape, namely that specifically for the onset, what you observe is a peak at 100 milliseconds, which is called the P100, and a valley at 140 milliseconds. So that's called the N140. I want to skip fast forward for a second because I want to check something. Yeah. Okay. So I thought this slide would come earlier. So I'm now talking you through the story step by step, and in the end you will get a summary. So I'm not presenting you the whole story first, and then present you the individual results. So we will get to the summary. So this is the basic response that you observe for checkerboards. Then if the checkerboard reverses, you get again the same response, but usually a little bit lower. You can see that already from kind of an adaptation perspective. First here, you don't see anything, and then a checkerboard flashes up, and then later on the checkerboard is still there and reverses, and you see a little bit of a lower response. These data that I'm showing you here, these are actually the data that you all have access to for your neurocognitive methods and programming report, where, as we discussed earlier, you can only focus on fMRI, you can focus only on EEG, or you can do both, whatever you like. If you do both, then you get the most out of it. So these are the data that we have there, but of course we were not the only one to ever record checkerboard responses. We actually did that because we wanted a very robust response for our EEG fMRI stuff. There is actually a clinical standard of how to record checkerboard responses. So these are also used in the clinics to test basic things of the visual system. And there is a standard, so you can download a paper that says if you want to record checkerboard responses, this is how you should do it, which is good because if things are standardized, then things also get easier. What is then also of interest in general for event-related potentials is that they, of course, are functions or modulated by certain aspects of the stimulus. And what we looked at here, and also which other people, of course, before it's looked at, is the contrast dependence. So contrast is essentially the difference between the bright part and the black part. Here, if you have really black and white, then the contrast is maximal. So this refers to a Michelson contrast of 1. Of course, you can make the white more grayish or light grayish and the black more dark grayish, and you get less contrast. And not too surprisingly, the responses that you get for lower contrast are lower. So the dashed line here shows the responses for lower contrast stimulus, which is kind of I would say is also if you look at that, maybe think twice if you want to get into consciousness EEG research, because in consciousness EEG research you will deal with stimuli that are really, really weak. And you see that, for example, for the reversal, the response is almost gone, even if you just have a low contrast stimulus, but you still have a stimulus, and the stimulus you can definitely see. So you can drive the EEG and the ERPs good with strong signals and not so good with weak signals. So if you are like me and you are not after the fancy effects, but you are after the fancy explanations for the robust effects, that's what I like more, because it's less dependent on luck and more on work, then you might think twice about EEG and consciousness research. Just a side note. Contrast is modulating visually evoked potentials. What is also modulating visually evoked potentials is actually the spatial frequency of the checkerboard. So if you have a different tiling, so if you have very big squares, you get a different response with respect to very fine tiles spatially, and there is kind of an optimal intermediate size. I mean, you can think of that if you have so if you have a very big tile, you just have a black stimulus or white stimulus, you still get a response to that. But you get a bit of more of a response if you actually have some tiles that are black and some tiles that are white. And then at one point it gets to fine grade, so that it actually looks more like a grayish thing, and then again the responses go down. So there is so here you have very spatial frequency, and you see that the responses are biggest here in kind of the intermediate area of spatial frequency. And of course, these responses, they are not that selective for checkerboards. So if you, for example, use black and white stimuli that have black parts and white parts and are kind of in spatial frequency terms similar to checkerboards, you will get similar responses. Yeah? So in general, you should always, if you do a cognitive study, for example, now that we are doing decision from experience more with EEG, where we, for example, present numbers, actually the first thing that we presented were also checkerboards, but now using more abstract stimulus material, of course, we know that we get, if we would just present a checkerboard, we get some response. So you should factor in somehow visual responses to stimuli, which can, however, vary depending on the stimulus material, as we have already seen. So what I've shown you here and also on this slide are kind of selected electrodes and maybe average over some electrodes. So it's essentially not what I recommend to look at if you have just recorded event-related data. So these are things that you find in papers or textbooks, because they are easy to, they are visually easy, essentially. There are different ways to look at event-related potentials, because usually you will not record from like 2 electrodes or 3 electrodes, but standard today is 64 electrodes, at least 32 electrodes, and I think this is actually from a 32 electrode system, rough estimation. Oh, is it 64? At least 64 or 32? Can't really tell. So how many are these? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10, it's more like 64 electrodes, I would say. Can I tell someone there? No, but I think these are actually from... Who has anybody looked at this data? It should be... Do we recall with 32 or 60?
Oh, it's definitely not 32.
It's not 32,
It
right?
looks like 64.
Right. So we recall with 65 electrodes in the UTF-9. Interesting. Yeah, must be, but also I think visually it looks more like 65. Okay, so what this shows here, this is an SPM visualization. So some people think SPM is bad for EEG. Other people think it's great for EEG. I think for the basic stuff, SPM for EEG is not that great. It uses field trip a lot, so there's not much original SPM actually for stuff, but specifically for visualization, it's maybe not the greatest ever. Still counting. So this shows all the electrodes for an entire post-stimulus time window, and so here time point 0 happened, and then here we are at 100 milliseconds, and you can see that in these dorsal electrodes you actually see the positive response that we just seen, but there are other electrodes which are more frontal where you actually get the negative part of the dipole, if you want. So you get a negative response there. So these, looking at selected electrodes, of course, does not fully portray what's actually happening. And we will see a different representation of that. This is something one can look at. One still somehow needs to define how to stack them so that one kind of gets an overall feeling. This is another SPM output that shows all the electrodes, and just by looking at that you see, so they are all scaled to the same degree, you see that something in the back, so this is the back, this is the front, something in the back on the right, there's more happening, and the reason for that is that our checkerboards were presented in the left visual hemifield. My personal, I think the easiest thing to visualize things is in terms of data matrices. So you have X on the time and you have electrodes on the Y, and then the values that you see in colors here are from each electrode at each time point the microvolt. And then you can see that there's a kind of a response pattern happening here. So there's something, actually a lot of negativity here at the P100, but also some positivity, as we will see in a second more clearly. And then the checkerboard reversal happened at 500 milliseconds, and then you see the response again. So I view event-related potentials then, so yeah, if you mainly like that, because this really describes the entire data, and especially if you're using something, if you're trying to simulate event-related potentials over the entire electrode space, which you can easily do because it's not like fMRI where you have really many voxels, but you have like 64 electrodes, then this is a good representation. But maybe it's not the most intuitive, and I think that really even more intuitive than this is what I show you next, because this really shows the dynamics of the whole thing. So now this shows again this data, typical visually evoked potential data for a checkerboard from a single subject that was presented with a left visual hemifield, and we are looking at here the traces of all the electrodes, and here what is called, I think, a topo plot most of the time. So what this shows is the arrangement of the electrodes that you have here in space. So these little dots here, these are the actual electrodes. And then you can, of course, spatially interpolate. So if you have, for example, you only have values from where the dots are, but then you can, of course, fill in what, so you can fill in the space between dots based on a interpolation. So, for example, weighting this by the distance between them, so that in the middle between two electrodes you have exactly the average value of the two electrodes. Of course, there are different ways to do spatial interpolation, so what you then get is kind of a view of the potentials as they are extended over scalp, and these views are always oriented in the way that you look from above, and the participant's nose is here and the back of the brain is here. Of course, there's a problem, because if you think about how these electrodes are on the head, they are on essentially a sphere, and you now look on a 2D projection of that. So it's a little bit not that easy to interpret what's going on with things that are at the borders, and especially things that are in regions of the space where things are quite curved. But you have a feeling for that because you've all seen the map of the Earth, which is also a 2D projection of a spherical thing called Earth. So this is what we're looking at here, and we're looking at a time point 0. I don't think there was any baseline correction, but there was definitely averaging. So anything that would have happened as random fluctuation over the trials of that has averaged out, and you see basically nothing is happening. And now we move through time. This is what I'm after, because the event-related potentials are things that happen over time. So they are actually spatial temporal fields. They are spatial temporal fields. So this is actually why I, in the EEG FMI integration work that I do, I use the theory of random fields and specifically spatial temporal fields to model responses of the brain, because these things extend over space and time, spatial temporal fields. So random fields are important if you want to do statistics on these fields. Now we go forward in time to 100 milliseconds, and you can see that there is positivity at 100 milliseconds in the contralateral side to the stimulus presentation. So the stimulus was presented essentially where the word checkerboard is written. And then, of course, because of the way, as you all know, the visual system is set up, most of that gets projected to contralateral visual cortex, and you get a positive response here. You get, of course, and this now relates to the bipolar activity, if you have some positivity and if you have a clear dipolar pattern. And in this thing we will talk about where the P100 is coming from, it's actually not primary visual cortex, but rather higher level visual cortex. If this dipolar activity is the only thing that's happening throughout the whole brain, and it is clearly arranged, then you get this strong dipolar pattern also in terms of the of what you see on the scalp. So this is basically a real data visualization of what I've shown you here. Yeah. So now the idea is that there's one dipole in the whole cortex active at this time point. So you get a projection of its poles to the scalp, and you see a clear dipolar pattern. There. Now, and it's because usually you would see the electrodes here, and you always think of the P100 as something positive. Yes, it's something positive, but on a lot of electrodes, the P100 actually looks negative, because the P100 is linked with the idea that we look at electrodes over the visual cortex, which are, of course, the electrodes here in the back, something like P08, O2 for the right side, and correspondingly P07 and O7 for the left side. Now we go forward in time and see now a more bilateral, although this of course definitely has a more of a focus on the on the right hemisphere, we see a negativity here and some positivity in the rest of the brain, but we don't have this strong dipolar pattern. Yeah? Because, of course, we also don't record the whole thing completely spherical. So, I don't know, I would be, can I guarantee that? Not sure whether I can guarantee if we have that here, that we would see a positivity, for example, on the chin where we don't sample, so I don't want to put my hand into the fires for that, but one could imagine that. But then also the problem is that activity that is not really the first activity, or in this case the P100, the second activity, is usually not of that clear of a dipole pattern. We have multiple dipoles that maybe cancel each other out, and you still see something, but it's not that clear anymore. So, so these clear dipolar patterns in the topography you see really for the early responses. So that's how that looks. This is okay. I already implied that you know what's going on, so I think everybody's familiar with this. I think this is always a nice representation of the visual system. And it's also not completely unimportant right now, because I have to tell you a little bit about the functional anatomy. Have you done that already with Adrian and next week? And you will talk about the visual system or general anatomy? General anatomy. Good. Like 2 years into your degree, maybe that's not that dramatic, but okay. The visual system, maybe but you learned about primary and so V1, you learned that in undergrad statistics, definitely not, but in undergrad psychology. So you know that the visual system is hierarchically organized, and it's kind of always the model system for the entire brain, which some people believe is very hierarchically organized. And you know that primary visual cortex is the first one, is V1, and if you look at it from the side, then it's at the calcarine sulcus. Again, you don't see this laser pointer, so it's here, the primary visual thing is primary to the inside, and then you can have other visual areas, V1, V2, V3, V3A, V3B, and then lateral occipital areas, which is kind of der ganze Stolz of the vision fMRI people, these different areas. But it's a nice model system to do things. Now, you need to be reminded of that, because the question is, of course, where do the different peaks in the event-related potential that one can observe come from? And the first response that you observe, we actually didn't observe for our checkerboard stimulation, because we presented a whole semi-field, so a whole half-field checkerboard. That means we covered both the upper and the lower visual field. And if one goes a little bit more into the anatomy of the visual system, one finds actually that, again from the Luck book, that the upper part of the visual field is projected to the lower bank of the calcarine fissure. So this representation here, this is a different way to draw what we see here. And the lower part of the visual field is projected to the upper bank of the calcarine sulcus. The idea is then that if what we did present both of them, then we have generated two dipoles, one being up and one being down, and the two of them canceling each other. So the first response that you can observe in a visually evoked potential for visual stimulus, the first response that we can observe from the brain is called C1 or N70, happens at 70 milliseconds. And the idea is that you can only get that if you have stuff presented to the upper or the lower visual hemifield, which is shown here, kind of with drawing. So if you have an electrode here, essentially like we had, then this will cancel out. If you if you present both of them, then this will cancel out. If you present only one, then you will only have one of these dipoles, and you will observe this C1 activity. Personally, I have not recorded that. There's some debate with Stefan Appelhoff right now whether we have a C1 response in the data that we recorded from Decision from Experience, and he's looking more into that. If you're into this and you want to do a master thesis on this, come in, and then we check whether we actually can also record these N70 C1 responses. By the way, I will say that again, but if you're interested in the whole random field theory for p-values or general random field theory, the whole thing that I'm doing in the other course, this is actually a research project. So come in and tell us if you're interested and work on that if you're interested. But I will give more specific pointers at specific locations. So this was something that we did not observe in our data, but it's really the first response of visual cortex to an event-related potential. What I've showed you are P1 and N1 responses. And then I would have thought that because EEG has been around forever and the ability to show flashes to humans has also been around forever, that it's been clear forever where P1 and N1 actually come from. Turns out that is not that clear. This shows data from a paper by Di Russo from 2002 in human brain mapping. So it's only from 2002, it's only 16 years old, that tried to dissociate using a model with multiple dipoles. We will go over this source localization and dipole modeling later in the course, but for the moment you can think, and this is what we will discuss later also, is that you can try to explain the data that you see on a scalp using dipoles, simulated dipoles that you put into a brain, and then basically simulate how the EEG response would look. This uses this dipolar modeling approach and, of course, EEG data to figure out what's actually going on. And there are 7 dipoles in this case, and one of them is located in the location of the primary visual cortex, so here in the middle. And then we have a P1 for 2, and early P1, late P1 for 4. And this is also then, I guess, Hemi field. So you see that the dipoles for 2 and 4, they are more, so you can also, of course, see them here. They are not where primary visual cortex is. Primary visual cortex, maybe I should make that a little bigger. Primary visual cortex is where the 1 is, and where the 2 and the 4 is, this is more lateral occipital cortex. So again, if you look at this is the nose here, this triangle, and these are the ears, just that you are clear about what you look at. So this indicates this study, and also other studies, but this is, I think, the study that I found communicates this most clearly, that P100 is actually generated by lateral occipital cortex, so more high-level visual areas already. So does anyone associate anything with LO? So what does LOC, the lateral occipital cortex, do?
Object recognition.
Exactly. That's the story. And so that's definitely not low level, because it's kind of looking at integrated stuff. Then the stuff that goes on for N140, that's even higher level and even more to the front. So that's even higher, more high level visual cortex, or going into parietal cortex, where you don't have these clear cut labels anymore. I mean, people try to establish these labels, but it's a little bit more iffy than V1, V2, V3, V3A, V3B. and so on. And this combined with fMRI data also basically points towards extrastriate. So extrastriate meaning is not in the striate cortex, so which is of course the calcarine sulcus, aka V1. So there are like 4 labels for the same thing: striate cortex, calcarine sulcus, V1, primary visual cortex. What's it? Area 40, 37? Actually, I don't know. Brahman areas. I stopped memorizing Brahman areas at one point. So there are like 5 labels for the same thing, which is not a good sign. Good. So that's C1 / N70. You can also see the strange labeling. So N70 is labeled by, I didn't say that actually, C1 for central response 1, and 70 for negative response at 70 milliseconds, and then P1 and N1, aka P100, N140, being generated by primary and higher level visual cortices respectively. And I think this is where we take a break and then discuss the modulation of these a little bit further.